Let's start. Welcome back, everyone, to the Genome Application Symposium. Our next session will deal with genomics of unconventional species, unraveling Earth's hidden biodiversity, and it will be co-chaired by Olivier de Clerck and Jean-Baptiste Ledoux. Go ahead. Thank you.
I'm not sure if I'm muted. No, I'm not muted.
No, you're not muted, and we're seeing your screen, but I'm not sure if it's in presentation mode. But we're seeing the slide.
You're seeing the slide? Okay.
Yes.
Good. So, yeah, welcome everyone for this session on genomics of unconventional species. So it'll be Jean-Baptiste Ledoux and myself, Olivier de Clerck, who will be presenting this and guiding you through the next couple of slides. Good. So I think if we've been both Jean-Baptiste and I, we've been looking around a bit at large genome sequencing initiatives, and indeed the ease at which genomes are sequenced nowadays, it translates into these large-scale sequencing programs, so the Earth Biogenome Project, and then of course Darwin Tree of Life, and then the ERGA European Reference Genome Atlas. Good. Now, this ease of generating genomes also translates into reference standards, higher reference standards for genome biology. For example, and these are some statistics that we copied from the Vertebrate Genome Project, which aims to make this benchmark for newly assembled reference genome. So you see N50, so the size based for context and scaffolds, it's larger than 1 megabase or 10 megabases respectively. Sequencing error should be very, very low. Structural variants should be confirmed, and then at least 90% of the sequences should be assigned to chromosomes or haplotype phased. Good. Now, of course, these higher standards, these go hand in hand with developments in recent sequencing technologies. Think about long-read sequencing, 10x genomics, or high-C, etc. And so the stakes are high, and the standards are basically increasing. Good, but in practice, and this is where the unconventional species actually come in, things do not always work as easily as according to the textbooks. So we'd like to delve into the next couple of slides. First of all, unconventional. So what is this? What does it mean to be an unconventional species? It can, in our idea, it can have different interpretations. You can be unconventional because of sampling, identification issues, sample access, quantity of tissue, the sample conservation to obtain good DNA from extractions, molecular biology, potential problems with high molecular weight DNA, but also the bioinformatics side of makes also that some organisms are unconventional. They can have very large genomes or genomes with a high number of repetitive elements, polyploidy, etc.
It's my turn now to speak here. And so we started a bit this to prepare this session, asking us, after several years of this large-scale reference genome sequencing project, what do we know about this unconventional species? So we did as it's usual to do in this case. We went to scientific papers, and you can go to the next slide. And to say the truth, we found there are all these big papers saying why it is important to sequence to have reference genome for all the species, but Olivier, please.
Sorry, that was one too much.
But they are usually wide scope, and the problem with unconventional species are almost to some point diluted in the paper. So from this scientific paper, I decided to go to check what we have in the GOAT, in the genome hubs. And here is a figure representing all the available genome assembly to date by phylum, class, order, family, genus, or species. And if you go on the next slide, yeah, we decided to check this part of the graph, which say that we now have assembly for species from 55 of the 67 existing phyla. So does this mean that the species for those 12 missing phyla are unconventional? So I look into the data, and these are the 12 missing phyla with no genome assemblies. And as you can see on the next slide, most of these species are very microscopic aquatic organisms for most of them. Okay, Olivier. Yeah. So if we take the other perspective, looking now from the species, we have assembly for 80,000 species over the more than 1 million species described to date. If we enter a bit more in the data set, in the database, sorry, we know that from this 80,000 species, only 23% of the species have high quality reference genome, which means assemblies at the chromosome level. So what should we think about the remaining 77% of species? Are those species unconventional species? This is the first question. What are the factors impeding to reach the chromosome level assembly in those 77% of species? And as a side question, maybe something that we can discuss further in this session, at the end of the session, during the discussion, is to know whether or not it is realistic to expect to obtain chromosome level assembly for all the species. Okay, so different factor impeding to reach chromosome level assembly. Olivier already mentioned most of them. So you have a molecular biology perspective, which is linked to species feature. For instance, the difficulty to obtain large amount of DNA. Many sequencing technology still need some relatively high amount of DNA. The quality of the extracted DNA, the IC library preparation, if you want to increase the scaffolding of your assembly, all this can be a difficult step to reach the chromosome level assembly. Then you also have the bioinformatic issue linked to different features of your genome, such as the size of the genome, the polyploidy, the amount of repetitive elements, or even the occurrence of many symbiotic species in your in the tissue sample you start from. So this is a brief overview of what we think when we start to prepare this session. And now Olivier will present a bit more his case study, and I will come back later to talk about octocoral reference genome.
Yeah. Thank you very much, Jean-Baptiste. So indeed, so we've had a general introduction on what it means to be unconventional, and I'll be presenting a project that together with Jessica Knoop, I will be conducting within the framework of BGE and ERGA, which is about genomic resources of Porphyra. So Porphyra is a red alga. You can find it in intertidal environments. It's subject to harsh conditions, lots of desiccation. Also, salinity changes are very profound And so here you see some, well, you see an aspect of what an intertidal environment looks like, and also these leafy, dark purplish to brownish seaweeds. So these are Porphyra. So Porphyra or at least a genus that was recently split from Porphyra is actually a major commercial crop species in Asia. Here you see some pictures of large-scale farming of Porphyra, and we know here in our well, I'm not sure if everybody's here from Western Europe, but let's say in Asia as well as in Western Europe, we know Porphyra best actually as this because it's used in the preparation of Japanese dishes such as sushi. Good. So what do we know about the diversity of Porphyra? And I would say that contrary to a lot of seaweeds, the diversity of Porphyra is reasonably well characterized within a European and actually also a global context. Here you see some of the species, Dioica, linearis, purpurea, which are commonly encountered. Now, Porphyra is definitely also an organism with a complex life cycle. So that already makes it a little bit more unconventional. So like many seaweeds, we have alternations between a haploid and a diploid phase. And in this case, the haploid phase is the foliose seaweed, while the diploid phase is termed conchocelis here. And this is actually a filament that lives usually inside calcareous structures, such as shells of bivalves, oyster shells, for example. And these two generations, these alternate. But we also see that there is a lot of asexual looping. So both the haploid as well as the diploid phases can display asexual looping. Actually, there is a lot of uncertainty regarding this asexual reproduction, whether all of these different spores, whether these entail shifts in ploidy between the various life stages. And indeed, then here I would come definitely to what I think is the most unconventional about Porphyra. I try to visualize it this way: if you have this conchocelis here, so it makes this conchospore, which is this blue sphere. Now, normally speaking, you would have you would have a meiotic division that yields 4 haploid individuals, and these 4 haploid individuals would be genetically different from each other, but not so in Porphyra. What happens is that the spore is released and it starts germinating, and in its early life stage, a meiosis occurs, which makes that these haploid blades are actually a chimera of 4 different haplotypes. And so if you sample these blades from the shore, so you always have genetic, you have a tissue that is essentially haploid, but which is chimeric in nature. Here you see this visualized in an experimental study by a Japanese researcher, Niwa, where he looked at color mutants. So we have color mutants in Porphyra. So we have a green and a red one, and you can clearly see where this banding, this is actually visualizes quite well this chimeric nature of these blades in this particular case. Now, a second reason or a third reason why Porphyra would be labeled as unconventional is because of reports of polyploidy and mixoploidy. This is mainly work by a Portuguese researcher, Helena Varela Alvarez, who's been looking based on flow cytometry data and microsatellite data on presumed ploidies, and she finds a whole array of different ploidies between different individuals, but also within individuals. So we have conditions of mixoploidy, for example, where part of the thallus can be 8x or 6x. And then the question, of course, is, I mean, from a population genetic structure, from a diversity perspective, how do all these organisms with different ploidies actually interact with one another? And this is actually what we definitely would like to address within our project. So how do these patterns of hybridization, ploidy, polyploidy variation, and mixoploidy translate into genetic diversity found in European Porphyra populations? And then, of course, how are these patterns actually related to life cycle variation and asexual reproduction? So what is available for us at present in terms of genomic resources? Well, there's one species, Porphyra umbilicalis. It had its genome sequenced and published in 2017, but so the genome itself is relatively small. We're talking about something smaller than 90 megabases. But when you look at the number of scaffolds, it's about more than 2,000 scaffolds, and an N50, which is very low here as well. So let's say this genome that is available to us is very much pre the standards that we try to use nowadays. There's a fourth species, Porphyra purpurea. Its genome should be in progress in the Darwin tree of life. And then for the other two species, Dioica and linearis, there is no genome project except for the one that is represented in this case study. And then we'll try to deliver this to the community of seaweed researchers to address this story. I'll pass on the word to Jean-Baptiste.
Maybe you can stop sharing and I will share my screen. Maybe it will be easier.
I will do this.
Thank you. So you can see my screen now? Yes?
Not at the moment yet here. I see a black screen at the moment.
Okay. And now?
Not yet. Yeah, it's okay.
It's okay. Okay, so let's move on the case of the octocoral. I will just briefly give you some phylogenetic context. So octocorals are cnidarians, and they are the sister clades of Hexacoralia, and both clades are within the anthozoans. The two clades harbor a huge diversity. They are found in almost all marine ecosystems, and they also share big conservation concerns, in particular due to a negative impact of climate change. However, if you check in the databases, we already reach almost 200 assemblies in Hexacoralia, whereas we are only at 24 different assemblies for octocorals. So here my point will be to show you why we should increase the assembly in those species using three examples, one based on species delimitation and biodiversity assessment, one on conservation, what genomic resources can can provide for the conservation of those species, and then I will develop unexpected outcomes from whole genome we released recently. So to start with the biodiversity assessment, this clade, as mentioned, is highly diverse with more than 2,000 species, which are found from shallow tropical seas to deep sea ecosystems. The taxonomy of octocoral is a bit tricky because there is no real diagnostic morphological characters. And on the top of that, the mitochondrial DNA barcodes are usually unsuitable in those species. You have different species, taxonomically different, which share the exact same mitochondrial DNA. In the recent year, there was a huge improvement in the phylogeny of the octocorals, thanks to the use of ultra-conserved elements. Yet, the problem of species delimitations remain, and I will now show you an example, in Unicella species. So this is a topic that was first addressed by a colleague of mine, Didier Aurel, who is from the University of Marseille. And this topic is that you have 3 different Unicella species in European water. So you have Unicella verrucosa in the Atlantic and in the Mediterranean Sea, Unicella cavolini and Unicella singularis, which are only found in the Mediterranean Sea. And Didier first started to study this kind of intermediate form that are found also in the Mediterranean Sea in population of cavolini or singularis. Using rat sequencing approach, he showed that those intermediate phenotypes are in fact hybrids between Unicella cavolini and Unicella singularis. So this is an example among few others in octocoral of fusispecies limit and hybridization between species from the same genus. So here, it's most likely that having good genomic resources can help to go further in this topic. Then switching on the conservation perspective, as mentioned, anthropogenic pressure and in particular warming-driven mass mortality are impacted very strongly octocorals. And this is particularly true in the Mediterranean Sea and in those 2 species, the red coral, Corallium rubrum, here on the left, and the red gorgonian, Paramyricea clavata, on the right. Those 2 species are habitat-forming species in biodiversity-rich communities, so they have a very important ecological role in their community. But since a couple of decades, you have a strong marine and recurrent marine heat wave in the Mediterranean. And you can see on this picture the effect of one of these marine heat wave. On the left side, you have one LC population of red coral in 2012, and one LC population of the red gorgonian in 2012 also, and the same population after the impact of the 2022 marine heat wave. So you can see here that almost all the red gorgonian are dead due to the extreme thermal stress. This kind of disastrous impact is observed recurrently, as I said, since 2 decades, and impact thousands of kilometers of coastal habitat in the Mediterranean. So there is currently a huge conservation effort from field ecologists to molecular ecologists and also a biodiversity manager to try to limit a bit the impact of this marine heat wave. Yet we still have numerous gaps of knowledge regarding, for instance, the local adaptation of the species, their demographic history, and the potential of genetic factor underlying a differential response to thermal stress. So here again, genomic resources are crucial to move on the conservation of those species. So considering this combination of the need for biodiversity assessment and conservation concern, we start developing genomic resources and whole genome reference in octocorals in 2017, I guess. And this is where maybe the octocorals are some kind of unconventional species, because these are the kind of DNA extraction we obtain at the beginning of this project. As you can see, highly degraded DNA, which hamper any long-read sequencing. And so problem on both sample conservation and high molecular weight DNA extraction. Our first attempt led to the first reference genome in Paramyricea clavata, which was published in 2020. And Olivier was mentioning a highly fragmented assembly just before for his algae, but you can see here it can be worse. We are here with a draft genome, highly fragmenting more than 60,000 different scaffolds. Since then, we made a lot of improvement, particularly on the DNA extractions. And you can see that we are now able to reach almost chromosome level assembly in some of those species. So just to mention that the three Unicella reference genome we recently generated are currently used to move on the biodiversity assessment and species delimitation study. And I will now focus on the reference genome we obtained for the red coral, Corallium rubrum. So this genome was obtained in the framework of the Catalan Initiative for the Earth Biogenome Project in collaboration with different laboratories, the CENAG and the Metazoa Phylogenomics Lab in Barcelona. And this genome, which is almost chromosome level, is the backbone of the case study supported by the Biodiversity Genomics Europe on the conservation of the red coral. I will go into detail of this case study, but I will show you how having this reference genome open a new perspective in my case. And to start this, I have to mention that octocoral and their ecology rely on secondary metabolites. They interact with their environment through secondary metabolites. In 2021, I guess, this paper was published showing that, contrary to what we thought, the gene encoding for the secondary metabolites were within the genome of the octocoral. So those metabolites were not synthesized by symbiotic microbes, but they were synthesized by the host, by the octocorals. So this was a very beautiful result. And following this, the same team, so which is led by Bradley Moore from the Scripps Institution of Oceanography, move on this work of the biosynthesis of secondary metabolites in octocoral. And Natalie Grayson, a PhD student of Bradley Moore, contacted me 1.5 years ago because she was interested in this part of the phylogenetic tree, and more particularly in five families of octocorals, which synthesize briaran terpenoid. And so she was currently assembling the genome of different species, and she asked me to share the genome of Corallium rubrum, which is one of the species synthesizing the briaran terpenoid. And of course, I was fully positive with this proposition. And she moved on and using, well, she did a huge work, but I will present only the result of the genome mining. She was able to show that all the species, including the Corallium rubrum here, harbor a biosynthetic gene cluster in their genome. So this can seem to be a small result, but you have to take into account that this is the first example of animals employing gene cluster family to produce specialized metabolites. The preprint is entitled like this, and you can find it on the internet if you are more curious about this beautiful story. So just to sum up on this, the development of genomic resources in octocoral was relatively challenging at the beginning, but I'm fully aware that it can be worse. And what I learned from this is that maybe when starting to work on such unconventional species, it is important to build a big team. It's a collaborative effort with people with very different skills, from taxonomists to bioinformaticians. And yeah, for sure, those kind of resources are improvement for biodiversity conservation and restoration, and you can always expect unexpected outputs. And that's all for me, so thank you. I don't know if you have any question, or if we can move to the discussion part. I have prepared some, like it's usual now since this morning, I have some Mentimeter question that I will share now to try to start the discussion. So I will put the code in the chat. Here it is. And I will share my screen for you to have the QR code. You see my screen?
Yep.
Yeah. So starting with the first question, we wanted to know whether or not you consider your species as unconventional. So it's
Yes,
quite
50-50,
balanced.
like that, yeah.
Yeah, yeah.
Mhm.
We have someone thinking that the question is not relevant. 30 people, maybe. Yeah, it's. I was expecting. I was expecting the much higher score for people considering the species as unconventional. It's funny. 10 seconds more. We only have 33 people. Okay, so it seems that most of you think that their species is unconventional, is not unconventional, sorry. I don't know for you, Olivier, but for me, it's quite of a surprise.
Yeah, it's yes, it's a surprise, and it's no surprise. I think definitely. I mean, there's definitely a lot of genomics of let's say terrestrial mammals and insects, I would think. Yeah. So therefore, I think these would lean towards the conventional species. Yeah. But I think the genomic public does draw a lot of interest as well from people that work on yeah, just weird groups that are at present still understudied.
Let's move on to the next question. So yeah, maybe also this question is a bit redundant with the previous one. Do you believe the unconventional species to be well represented in the current reference genome projects?
This is a different question from answering whether the current reference genome projects are adequate tools to generate high quality genomic sequences. It's just about the representation.
Yeah. Well, in fact, when you check the databases, there is a lot of unconventional species that are currently in progress. But it seems that some of them get stuck since quite a long time in the same status, you know, when you can see the status of sampling, sequencing, assembly, etc. And many of them are still stuck in the sampling stage.
Yeah,
so here we have here we have a huge majority of people thinking that unconventional species are unrepresented in the current reference genome project. Okay. I will move on the next slide. So what makes a species a species unconventional? Its ecology, including the ease of sampling, molecular biology, lab work like DNA extraction, library prep, or only the bioinformatics stuff. There is no...
I find this very interesting. This morning was also a session where quite unexpectedly sampling showed up to be a very important aspect in these genomic processes. Yeah. So
yeah, but I think sometimes maybe those very preliminary but important step like sampling, species identification, and sample conservation, I think sometimes they may be a bit of overview by the in this project, in the mean that we now have the sequencing technology, so we are able to sequence everything, but it's not so clear. I mean, if you don't have someone to clearly recognize the species, if you don't know exactly how to extract the molecular weight DNA, you can have all the technology you want after. It will always be a problem. I don't know.
True. But definitely the, let's say, DNA extraction, library prep, I mean, does come out as a first, as the most important hurdle. And I mean, from practice, working on seaweed, this is really very difficult, especially when you move from short read to high to long read sequencing. Yeah, it is. It can be very depressing sometimes.
Yeah. So we have some comment in the chat from Camila, saying that science, in particular connected to conservation, is still very biased toward conventional species, and that's true. Yeah, that's true. I guess. Yeah. And in BGA, we are handling a high number of unconventional species. I know it, and Olivier too, by the way.
We're very happy with
Not
this.
easy, yeah, for sure. It's a great opportunity.
Absolutely.
Conventional. Alessia Cariani said that conventional species concept might vary between freshwater, marine, and terrestrial rim. And Zofia adding that it's probably also vary between the wet lab scientist and the bioinformatician. Yeah, it's true. Maybe Alessia, I don't know if you want to expand a bit what you mean.
Here I am. Yes, because maybe people working in the marine environment have to face a lot also the three-dimension is an environment which is more three-dimensional. So like sampling octocorals may have also other challenge like going into the more deep environment or environment in which directly operator cannot go.
Yeah, true.
Maybe this is the same. This is the same also for other very extreme environment like high in the mountain or.
Yeah, for sure. The ecology of the species is an important characteristic of the unconventional aspect. So I still have one more question, I guess. I will come back to the chat later. Yeah, so Olivier, what's holding you from sequencing your unconventional species?
So it's not the technology, it's not the difficulty of getting high quality DNA or enough DNA, etc. But I think Jean-Baptiste included also collaboration because, let's say, for many of these less well-studied groups, the scientific communities that are involved in studying such organisms are also much smaller. But it doesn't appear to be the...
My idea also with collaboration is to emphasize that, to my point of view, or to my, with my brain at least, let's say, it's not, it's very difficult to be able to sample, to be able to know how to fine-tune a DNA protocol extraction. I mean, at least I was not able to be on all the front at the same times. So having this collaboration with people with a lot of experience in molecular biology, for instance, for the IC library preparation, I work with people from Rosa Fernandez's lab in the Metazoa phylogenetic lab in Barcelona. And it was a huge benefit for the project. I would never have been able to do the IC sequencing on those species without their knowledge and their help in this topic. The same is true with people from the CIIMAR that were also here to assemble the genome. So this was in this sense.
I think perhaps collaboration and finances could be, I mean, to some extent, these are linked, I think. Like groups that attract a lot of researchers usually have large consortia. So lots of collaborations, which can make it easier to leverage the necessary funding and get your project funded. Yeah. So I think the smaller consortia, may be disadvantaged financially.
Yeah, for sure.
Yeah. And especially if then these less studied organisms then tend to have difficult genomes, very large genomes or full of repeats, etc., then it becomes a very difficult task to become successful with your genome project.
True. And do you expect a technological breakthrough allowing quick chromosome level reference genome assembly in any species in the next 5 years, in the next 10 years, maybe one day? Or do you think this is a quest without end, let's say?
There's a lot of optimists in the
yeah, which is nice. It's nice, yeah.
Or perhaps the PhD students that have to defend their PhD within the next 5 years.
I don't know if some people have comment on the question. Please let us know.
But we also have two people who are downright pessimistic. It's like it's not going to be a solution.
Yeah. Maybe 10 seconds more.
Yeah.
Some more people want to add something. Then the last question, just to know where all these optimistic people came from, or what they study, to say it. If they are studying terrestrial plants, fungi, aquatic plant vertebrates, aquatic invertebrates, protists, or terrestrial invertebrates. And yeah, I'm expecting the vertebrate to rise because it was too optimistic. So yeah. Yeah, majority of people working on vertebrates.
Yeah.
To some point, it's not so surprising with the previous.
So perhaps a workshop where you expose people that work on vertebrate genomics to marine invertebrates or algae. Yeah.
Or algae, because I think
and to get reasonably high quality DNA out of it.
Yeah, can be a good idea.
Yeah, no.
So yeah, 13 people on vertebrates, Well, aquatic plant, Olivier, there are still a lot of things to do. Huh? Not yet.
I did both.
Not to mention the fungi. Nobody is working on fungi. Okay, so that's all for the Mentimeter.
Okay.
I will stop sharing my screen. Up. And I don't know if someone has some question or comments. Gabor is asking, is it unconventional if my plant is diploid, but the haploid genome size is 10 gigabytes?
Yeah. I would say good luck.
Yeah, I would say, yeah, it's kind of unconventional, but yeah.
It definitely increases the cost of whatever you want to do, because the sequencing effort you will have to go for, it's
and
it's not going to be a cheap project.
And the bioinformatician after that will have some work also to do, because to deal with such a amount of data.
I think in the interest of time, I think we should Rob Waterhouse raises a hand.
Yeah, just a very quick one. So it was very nice to hear about your positive experiences of talking with the sequencing centers, talking with others who are dealing with similar problems to find solutions. And really just to reiterate here in front of this large audience, so that's one of the main goals of the ERGA committees is to act as these kind of centers of excellence, or a forum, if you like, where the community can come together and discuss what was not working for them, what failed, what they tried 10 times and it still didn't work until they found a solution. To share then, where there have been some success stories in terms of finding the solutions to some of these difficult cases, if you like. And it's about, sharing the failures, but also the successes in order to make it easier for everyone to achieve the goal, which is a high quality reference genome first time round without having to waste lots of time and resources on troubleshooting issues that other people have already found some solutions for. So just to reiterate that, this is really good discussion here, and it's exactly the kinds of things that we would like to see also brought to the ERGA committees so that they can be shared more widely.
Okay, so this acts as an online forum where people exchange information?
Exactly. So, we have the sampling and sample processing committee. We have the sequencing and assembly committee. So I guess those are the two most relevant here, depending on whether the issues are happening at the sample and extraction side of things, or whether the issues are happening in the library prep and the sequencing side of things, or even downstream in the assembly, if you've got, super complicated reads that are coming out. And really, it's just to reiterate that the committees meet once a month, and everyone is welcome to join. Drop a line to the steering committee to say, look, I have been working on this problem for the last few months. My PhD student is going crazy. Can I have a 10-minute slot in the agenda to present my case and see if anybody in the audience has some ideas to help me solve the problem? So that's, for SSP, Sampling and Sample Processing Committee, but also for the Sequencing and Assembly Committee in particular.
I think this is very welcome information. Thank you very much. I think we should give the floor to the to the next presenters, right?
Yeah. Or I don't know if it's a break or
yes. Thank you very much, Olivier, Jean-Baptiste.
Anyway, thank you. Yeah.
So we will have a break now, and we will come back at 3 p.m. with the merits and limitation of reduced representation and low coverage analysis, genomic analysis, with João and Gabor. So
thank you very much.
Have a quick break and we'll come back at 3 p.m. Thank you, Olivier and Jean-Baptiste.
Thank you.